Hello and welcome to the immuno-oncology session of Advancing Science. I'm Peggy Sotiropoulou, R&D Director in Celiad, and I will accompany you in this track. Clinical activity against cancer has seen a major shift to immuno-oncology over the last few years, including areas of immunology that were not anywhere near mainstream thinking just a decade ago. As illustrated in this graph, published in Nature Reviews in Drug Discovery, the immuno-oncology therapeutic agents under clinical testing have almost doubled between 2017 and 2019, a fact that is also reflected by 31 new FDA approvals during this two-year period. Immuno-oncology is clearly in a time of strong development. Initially, antibodies paved the way with paradigm-changing treatments, while recently there is a big interest in cell therapy, with the CD19 CAR-Ts certainly impacting the global hematology practice. Importantly, with the increasing understanding of molecular immunology, the field is now expanding, including revisiting concepts that have failed in the past, such as cytokines, NK cells, disease, as understanding the complexity of the system allows tackling the reasons for the original failure. Summarizing the different areas being explored today, we could group the immunotherapeutic modalities in oncology in 6 groups. Monoclonal antibodies that can target only tumor antigens or tumor antigens and antigens on immune cells, tethering them for cell killing. Checkpoint inhibitors, which will release the brakes hindering host immune responses. Therapeutic vaccines that are targeting and boosting the immune system. Adoptive cell transfer, autologous or allogeneic. These ones will have the focus lately in CAR T cells and engineered TCR T cells. Oncolytic viruses that can kill cancer cells but at the same time activate anti-tumor immunity. And the recently emerged microbiome, which have been proposed as therapeutic, prognostic, or diagnostic tool. Of course, there's still a lot of challenges that we have to tackle, such as enabling the increased efficacy and reduced toxicity of the immunotherapeutic approaches, and we can do this by understanding the complex network of immune pathways and the role of distinct immune cell subpopulations, to understand the patient responses through basic and translational research, being able to predict the patient responses by developing reliable prognostic biomarkers, enable solid science by developing improved models to better understand the underlying mechanisms of the outcomes of tumor immunotherapy, and implementing the new technology, machine learning and artificial intelligence, to be able to integrate the real-world data and the single-cell omics data to recognize specific patterns of immune responses. Without further ado, I would like to introduce you to the agenda of this meeting. We have 4 speakers, Peter Nelson, Sergio Rutella, Juras Adamic, and Jenny Croning, that will talk to us about the landscape of the immune microenvironment in metastatic prostate cancer, for the correlation of immune infiltration with p53 mutational status in a multi-cohort acute myeloid leukemia, understanding the molecular and immunologic profiling of cytotoxic T-cell responses in patients receiving engineered DC vaccine, and finally, find what GeoMx platform can offer us with fully validated antibodies to meet the spatial profiling needs in every laboratory. Our first speaker is Peter Nelson, Professor of Medicine at the University of Washington, who will present the spatial landscape of the immune microenvironment in metastatic prostate cancer.
Hi, my name's Pete Nelson from the Fred Hutchinson Cancer Research Center in Seattle, and I'm going to be speaking about some work that we've done focusing on understanding the spatial landscapes of metastatic prostate cancer with a particular focus on the immune microenvironment. And this analysis was really made possible by using the digital spatial profiling technology. So I represent a large research group, as shown on this slide, with collaborators at the University of Washington, at the Fred Hutch, as well as at NanoString. These are my conflicts, none of which really have direct bearing on the discussion that I'll provide here. So this is an outline of what I'll focus on. First, just to lay the landscape out of why we're focusing on advanced prostate cancer. I'll describe some major research directions, the study design for the aspects of digital spatial profiling, and then get into two areas of results. First, focusing on the heterogeneity that we observe in metastatic prostate cancer, and then a dive into immune responses within these metastases, and then I'll end up with a few conclusions and future directions for next steps. So prostate cancer in 2020 is a highly lethal disease with more than 30,000 deaths a year. These are essentially all attributed to metastatic prostate cancer. You can see on this slide, bone is a very dominant site, as shown by this nuclear medicine bone scan. Essentially, when you see metastatic prostate cancer such as this, it is currently incurable. So how can we approach mortality? We think about this in four ways. One, understanding the drivers of the metastatic process and survival in advanced disease. We'd like to apply specific therapies, the whole concept of precision medicine, anticipating and understanding resistance mechanisms, and then co-targeting these anticipated resistance mechanisms. So we've deployed many therapeutics in advanced prostate cancer, including immune-based therapies, which I'll get back to. Overall, immune-based therapies, the typical checkpoint blockade inhibitors, have not worked well in prostate cancer, with the exception of a very rare subtype of hypermutated disease due to mismatch repair. We now recognize that advanced prostate cancer has numerous subtypes. These can be defined genomically. So this is a study from the Stand Up to Cancer Dream Team profiling metastatic prostate cancer. What you can glean from this is that there are many recurrent genomic events. So each row here is a different genomic event. Each column is a different tumor or metastatic site. Androgen receptor right at the top is the most frequently altered genomic event in advanced prostate cancer, and you can see that there are many others that are either frequent or infrequent. The panel on the right compares these genomic alterations in localized prostate cancer, that's the y-axis, versus the alterations in metastatic prostate cancer on the x-axis. You can see that certain events are clearly enriched in metastatic disease. We now recognize, after we apply therapeutic pressure, most of this focuses on targeting the androgen receptor, that we're now seeing resistance mechanisms that are phenotypically defined, often by the development of transdifferentiation to phenotypes such as neuroendocrine prostate cancer, a stem cell phenotype, mesenchymal, basal cell, etc. So in addition to the underlying genomic events, we now see phenotypic events, some of which are clearly related to an underlying genomic event, but others we still don't understand the process. So this is some recent work looking at the phenotypes based on gene expression. This becomes relevant as we get into the digital spatial profiling that I'll talk about in a moment. What you can see here is that we can categorize metastatic prostate cancer based on androgen receptor activity and neuroendocrine activity into, at least in this case, 5 phenotypes based on gene expression. So we have androgen receptor high, neuroendocrine low, those are the green bars, androgen receptor high, neuroendocrine high, or amphicrine tumors in red, androgen receptor low, neuroendocrine low, in purple, androgen receptor low, neuroendocrine negative in blue, and then the androgen receptor negative, neuroendocrine positive in yellow. So our major research and clinical questions relevant to this presentation, one is, what's the extent of inter and intra tumor heterogeneity with respect to these phenotypes in metastatic prostate cancer? And then second, why is prostate cancer essentially refractory to current immune-based therapies such as immune checkpoint blockade? So the first, could this heterogeneity impact therapy resistance? And then the second, are there other immune targets present in these metastatic tumors where we may redirect future therapies and studies? So our study design, we really wanted to understand inter and intra-tumor heterogeneity using digital spatial profiling, and then also determine if there were particular immune features of metastatic prostate cancer that we could also identify. So our study design is as follows. We used tumors that we acquire from a rapid autopsy program developed by Colin Morrissey at the University of Washington. In this case, we looked at 28 patients. We looked at 2 metastases from each patient. These were arrayed on tissue microarrays where we obtained 3 cores from each of these tumors. So we in total have 178 samples. For this study, initially, we took 1 ROI or region of interest from each of these arrayed TMA cores. Overall, because prostate cancer metastasizes to different sites, we were able to sample 16 different tissue sites, such as bone, lung, liver, and lymph node. We used the GeoMx DSP workflow, which I believe other presenters have described, so I'm not going to get into a lot of detail here. Basically, the approach uses for protein detection an antibody that's tagged with an oligonucleotide barcode, or for gene transcript expression, an oligo targeting a particular messenger RNA or cDNA, where you have that also barcoded. You then choose regions of interest microscopically and use UV light to cleave the barcode, which is then aspirated and dispensed into microtiter plates, and then you can either sequence it through next-gen sequencing or through encounter assays. In this case, from each TMA, we took 3 sections, the first for H&E and immunohistochemistry, the second for digital spatial profiling using RNA. This sampled about 2,100 genes with multiple probes per transcript, so about 18,000 probes. And then for protein, we looked at 60 antibodies that were also tagged. Okay, so the results of this study. First, inter and intra tumor heterogeneity across these metastases. So the first question we had is, does digital spatial profiling classify the prostate cancer phenotypes that we had previously identified using RNA-seq-based bulk tumor analysis? So this slide's a little bit complicated, but the color coding is as follows. At the very top, we're looking at the different sites for metastases. For example, in red, that would be a bone metastases. Then we have a series of gene expression signatures indicating a pathway or a program activity. So we have androgen receptor, neuroendocrine activity, CCP, which is a cell cycle progression score, a surrogate for proliferation. And then the fourth is for FGF and MAP kinase signaling, which we've been interested in as a therapeutic target. Then the line below that shows the class determined by bulk RNA-seq. And then below that is the class that we assigned by digital spatial profiling using the nanostring technology. So you can see that, first of all, DSP-based classification clearly separates these tumors into the different phenotypic categories that we previously had determined by RNA-seq. You can see that shown on the upper right PCA plot. Below that, we've compared the androgen receptor signature of activity and the cell cycle progression CCP score, comparing the DSP-based result with, and that's on the x-axis, with the RNA-seq-based result for that same tumor on the y-axis. And you can see very high concordance here. So this really confirmed our impression of the accuracy of DSP-based classification of these tumor samples that was at least equivalent to the RNA-seq-based bulk sequencing. Importantly, we were also able to sample bone. So this has been traditionally a very difficult tissue site to work with because, for example, cutting sections or processing this often requires pretty severe decalcification processing steps. So these bone samples did go through our traditional decal procedure. Overall, the signal intensity or the signal for each transcript was generally lower in the bone sample compared to the soft tissue. However, it was readily sufficient to classify each tumor based on its phenotype. And also, we were able to find bone-specific features. If you look at the far right panel, this is a soft tissue metastasis and a bone metastasis from the same patient, comparing the two. And we can see that there are certain bone-derived factors, particularly found in that bone sample. So this is an attribute for this approach. I would also say that, I didn't mention it earlier, but all of the DSP-based gene expression studies were done on formalin-fixed samples dating back up to 10 years. The RNA-seq was done on frozen samples. So again, using the formalin-fixed is also a major asset in this type of assay, and allowing you to use retrospective samples that may not have been collected in a frozen state. So the next question was, what's the extent of intra-individual or inter-tumor heterogeneity? So in this case, what we're doing is we're looking at the multiple tumors from each individual. So we've shown the patients at the top. As I mentioned, each patient had 2 tumors at least sampled here. And so you can see the particular site of bone, liver, lymph node, lung, or other soft tissue. And then those gene expression programs that I described earlier around androgen receptor, neuroendocrine, et cetera. And then the classification based on DSP. So you can see that most of these tumors within a patient did have the same phenotype determined by DSP. However, there were some tumors here that were clearly distinctly different. So you can see, for example, patient 12011, the second patient over from the left, had one tumor classified as amphicrine or AR positive, neuroendocrine positive, and another tumor classified as AR positive, neuroendocrine negative. So generally, concordance across metastases within a patient, but clearly some outliers or some divergence in that. And here's an example of one such case, 15069. So here are 2 different metastases. You can see that their AR signaling or androgen receptor signaling is highly concordant within these 2 different metastatic sites, but the neuroendocrine signaling, as shown on the bottom panel, is clearly divergent. So these tumors classified phenotypically distinctly. So the next question was, what's the extent of intra-tumor heterogeneity? So this is where the spatial profiling can really shine, where the bulk RNA-seq failed to provide this level of discrimination. So now what we've done here is taken all of the tumors, and I'm showing you the individual regions of interest. So I mentioned for each tumor, we sampled 3 different ROIs from within each metastatic site. So that's what is shown here. So for each patient, we're looking at 6 different ROIs, 3 from each of those tumors. So we're starting to identify here that there is some degree of intra-tumor heterogeneity with respect to gene expression here. Here I'm showing you one example, patient 17017. where if you look at the far right at the top, one of the ROIs is distinctly different in terms of its expression compared to the remainder of the ROIs. So we were starting to get some idea that there may be some intra-tumor heterogeneity here. So by bulk RNA-seq, again, if we look across the two tumors from each patient, there was an 82% intra-individual concordance. That is, those two tumors from each patient had the same phenotype called, and those were boxed. However, if we look at the individual ROIs from those tumors, that intra-individual concordance drops substantially to 64%. So that 10 tumors here were discordant when you started to go and look at the individual regions within those tumors. So I'll highlight one particular patient here in red, 15096. So in this case, we had an impression that there may be heterogeneity within this particular metastases. So what I'm showing here is the simply H&E section, the full face section of this metastasis, where you can clearly see tumor regions within this particular metastatic site by immunofluorescence, really, for pan-cytokeratin, CD3, and DNA. You can see that there appears to be some diversity within this metastases. So we took a deep dive and sampled approximately 18 regions of interest across this different tumor. And what I'm showing you on the bottom are several of those ROIs pretty much marching across the face of this tumor, ROI 7 being right in the middle of that pan-CK positive green region. Then you start losing the pan-cytokeratin as you go across. And then the digital spatial profiling on the right shows that at the ROI within that cytokeratin positive region, you have high androgen receptor activity as read out by AR-regulated genes such as TARP and TMPRSS2. And then as you go across the face of this tumor into the more distant regions, you lose androgen receptor signaling, and you actually gain neuroendocrine gene expression activity. And you also gain MAP kinase signaling and FGF expression, as you can see by the middle and lower panel. And we know clinically that we need to treat these tumors distinctly differently than an androgen receptor active tumor. So the bulk RNA-seq from this tumor suggests that there was an amphicrine tumor, that there were AR high plus neuroendocrine high. But once you dig into the heterogeneity within it, you see that there was actually not amphicrine per se. There were regions that were AR positive, neuroendocrine negative, and other regions that were AR negative, neuroendocrine positive. So another question that we wanted to address was to actually look at splice variants. It turns out the androgen receptor has a number of splice variants that promote resistance to typical androgen receptor-directed therapy. So within the transcript-based digital spatial profiling, we designed a series of probes that could distinguish the different androgen receptor splice variants. So this slide is simply showing you that there are many different splice variants due, for example, to the inclusion of cryptic exons. I'll show you that in more detail in a moment. And a New England Journal paper showing that one of these splice variants, ARV7, again promoted resistance to androgen receptor targeting. So here's the structure of the androgen receptor. It's comprised of 8 canonical exons. And it turns out with the splice variant, in this case ARV7, there's a cryptic exon 3, that ends up being spliced in, and you lose expression of the more distant exons here. So what we did is design probes used for DSP that would capture cryptic exon 3 and we can compare that with a ratio of exon 4 to derive whether or not a tumor does or doesn't express ARV7. So here's the analysis. If we look across all of the tumors across these metastases and the individual ROIs, if you look down at the second line from the bottom, this is the ARV7 level assessed by RNA-seq, just bulk RNA-seq from the tumors. So you get an idea that most of these tumors either express ARV7 or they don't. However, by digital spatial profiling, you see much more heterogeneity within the tumor with respect to ARV7 expression. So ongoing work is designed to determine, does this degree of heterogeneity contribute to resistance to AR-targeted therapy that may not be apparent from sequencing bulk tumor? This shows you the regions of interest, for example, for this particular case that I'm showing, 12005 and 12011, and the diversity of ARV7 by digital spatial profiling, which wasn't apparent by bulk RNA-seq. Okay, so the last section here focuses on immune response in prostate cancer metastases. What we started off by noting is that if we look at markers that denote particular immune cells, so T cells, B cells, and even subtypes of these, is that the vast majority of these metastases really lacked any substantial immune cell infiltrates. Now, the caveat here is that this analysis is entirely focused on intratumoral regions of interest. We are not, in this case, looking at the peritumoral region or region in the stroma. So again, with notable exceptions, there were very few tumors with any substantial intratumor inflammatory infiltrates. And interestingly, we also saw that there was high consistency with any particular immune cell marker across the multiple ROIs from the tumors from the same patient, regardless of the metastatic site. So for example, if you had a metastases in lymph node versus bone, you basically saw the same immune cell repertoire. So again, with some notable exceptions, these were pretty devoid of cells. The two highlights that I have here, one shows a T cell response in all of the ROIs across metastases, and then the other orange highlighted tumor shows multiple different cell types, immune cell types within the two metastases from that patient that are distinctly different than the rest of the patient metastases. So the other notable thing is that if we order these tumors not by patient, but by phenotype, so in green are the AR active, neuroendocrine low, et cetera, on the left, and on the far right, we span all the way through to the double negative cases, AR negative, neuroendocrine negative. So you can see that there are some features that are immune cell features that associate with phenotype. Generally, the macrophages are present in the androgen receptor negative or low subtype that I've circled here in orange. We also looked at cytokine programs and immune markers and found that these are also conserved across the multiple metastases from a given patient. I won't go into all the individuals ones here. You can view them on your own on these slides. If we then order these by phenotype rather than patient, we can also see that cytokine programs and immune markers associate with the phenotype of the prostate cancer beyond simply the different patient sites. So what's the composition of these? So of interest, the AR negative neuroendocrine tumors and the AR negative neuroendocrine positive tumors are the ones with some intratumoral immune infiltrates, But the garden variety prostate cancer, those that are androgen receptor positive, neuroendocrine negative, are immune cold. Here we can see that in these AR negative, neuroendocrine negative tumors, there's high CD8B and PRF1, which are associated with cytotoxic T cells. Also TAP1, which is involved in antigen processing, possibly associated with attraction of T cells as a hypothesis here. And then in the stromal region, this is an example where we did look at the stroma. We can see that there are CD68 positive monocyte or macrophages is the predominant immune cell type in the tumor microenvironment for these metastases. We also see high levels of peritumoral CD163 positive cells, which is a marker of pro-tumorigenic macrophages. So the DSP technology also allows you to profile protein, as I mentioned. So we looked at, in this case, I'm showing you 57 of the 60 antibodies that were profiled. We used the same tissue microarrays that we use for the DSP of transcripts. So it provides another level of confidence, number one, of the transcript measurements, and also can determine other biology that you may miss simply by transcript levels. So importantly, we were able to show that if we compare the DSP-based transcript measurements with the DSP-based protein measurements, there was very high degree of concordance. I'm just showing you androgen receptor and a proliferation marker, and then synaptophysin, which is a marker of neuroendocrine tumor. So these were, again, highly concordant for those expected AR active associated neuroendocrine associated markers. So we also showed that androgen receptor measurement by protein did associate with those tumors that we classified as AR high. Also, the neuroendocrine marker CD56 or NCAM is high in the neuroendocrine tumors, as is synaptophysin. So those are the tumors on the far right, kind of shown in the yellow circles. So I mentioned earlier in the discussion that prostate cancer is highly resistant to immune-based therapy. It turns out by this analysis, very few tumors expressed any sort of signal for PD-L1 or PD-1. CTLA-4 has also been evaluated in prostate cancer as well as melanoma. We found very low levels of CTLA-4 checkpoint in these metastatic sites. Very interestingly and unexpectedly, we found very high expression of B7H3, another immune checkpoint molecule. Here we're comparing on the far right the transcript-based and the protein-based measurements of each of these checkpoint molecules, confirming very low expression of CTLA-4, PD-1, and PD-L1, and very high expression of B7H3. So B7H3, or otherwise known as CD276, does have some role in inhibiting T cell function. It has a variety of other involvement in the immune system, as well as promotion of tumor growth. It's expressed on a variety of tumor types. This cartoon on the left is a very nice review describing B7H3. We found B7H3 was very highly expressed on the AR active, AR high tumors, but much less so on the neuroendocrine and AR null tumors. And this coincided with the expression of another immune checkpoint, TIM3. So this is a deep dive into one of the tumors where we looked at multiple regions of interest. The vast majority of these ROIs expressed very high CD276 and TGF-beta. However, there were a few regions that had lower level of expression. So ongoing work is designed to try to understand this heterogeneity and determine if CD276 or B7H3 may be a reasonable target, specifically in prostate cancer. So I'll conclude here. Just to recap, I tried to show you that castration-resistant metastatic prostate cancer comprises many subtypes. Some are defined by genomic alterations, some by phenotype. The DSP-based profiling, spatial profiling, really demonstrated very high correlation with RNA-seq-based methods to define prostate cancer subtypes. We found profound intratumoral heterogeneity that RNA-seq had missed in several tumors. The use of formalin-fixed paraffin-embedded materials that had been arrayed on tissue microarrays was a major advantage for rapid throughput. And then we did also show that DSP-based protein quantitation correlated with traditional immunohistochemistry and allows you also to multiplex. We also showed that these subtypes can coexist within and across metastatic tumors from the same patient. And notably, very few inflammatory cells within these tumors of any type. The most predominant being macrophages. The vast majority had no CD8 or CD4 T cells. We also found very limited expression of immune checkpoint molecules where FDA-approved therapies have been used, which may explain the lack of treatment efficacy in prostate cancer and nominate a new immune checkpoint target, B7H3, that associates with the most common phenotype of advanced prostate cancer that expresses androgen receptor activity. So the future directions center on determining if the heterogeneity in phenotype, as has been shown for genotype, associates with treatment outcomes. We also want to test therapeutic strategies that co-target coexisting phenotypes, and defining this may best be done by spatial tumor analysis. We also suggest that B7H3 may be a reasonable target for immune checkpoint blockade and trials in advanced prostate cancer. And then finally, we'd like to further determine associations between the phenotypes and the tissue microenvironment components by also evaluating the peritumoral and stromal regions using spatial profiling. So we have many funding sources that supported this work that I've shown here, including the NCI, Prostate Cancer Foundation, Department of Defense, as well as support from NanoString. And then many collaborators, first and foremost, acknowledging the patients and families that participated in the rapid autopsy program, number of investigators at Fred Hutch, the University of Washington, and at NanoString. So I'll stop there and be happy to answer questions. Thank you.
Are there known drivers of tumor plasticity and phenotype heterogeneity?
So currently, we don't know the specific drivers of tumor plasticity and heterogeneity, but there are clear associations. For example, the ability for a prostate cancer to transdifferentiate, that is, move from one phenotype to another, plasticity, underlying that is loss of two tumor suppressors, retinoblastoma, RB1, and P53, and PTEN loss probably plays a role as well. These are more permissive to allow for plasticity in the context of repressing androgen receptor signaling. So a lot of work in the field is trying to understand this more stem-like state that's conferred by loss of P53 and RB that mirrors what we see in small cell lung cancer as well. So I think it's a very exciting and important area of work to see if we could prevent this transdifferentiation.
What are the microenvironment features that contribute to therapy resistance, and could they be assessed by DSP?
So there are a number of microenvironment features that we think may contribute to treatment resistance. For example, we know that most of the therapeutics that we use to treat both localized and advanced cancers are not exclusive for damaging, if you will, the tumor cell. They also have effects on the microenvironment. A number of studies have shown that damage responses in the microenvironment, usually due to genotoxic stress, DNA damaging agents like radiation, platinum chemotherapy, and others, can induce a secretory program from that microenvironment. These include Wnt signaling factors, much regulated by NF-kappa B. It also influences the immune cell and the immune environment, which in some cases could promote therapy resistance and tumor growth. So these microenvironment features are ideally suited to be assayed by digital spatial profiling.
Can you compare and contrast the DSP analysis of transcripts versus protein?
Right, so comparing assay of transcripts and protein by DSP, one of the major advantages of DSP is essentially within the same tissue sample or the same tumor sample, you can acquire both protein information and transcript information. So the transcripts currently are allowing for many more genes to be assayed. You can certainly multiplex proteins. In the data that I showed, we're looking at about 60 proteins. One of the challenges that each antibody has to be tagged specifically and worked up to show that it's going to perform exactly the same way as a non-tagged antibody. So some of the preservatives need to be accounted for in terms of the antibody. But clearly, protein levels don't always equate to transcript levels, and there are certainly things that we're missing at the transcript level that may be altered at the protein level. So I would say the major difference currently is being able to sample many, many more genes by the transcript-based assays, potentially the entire transcriptome, whereas proteins at this point require a good antibody, but using both assays simultaneously certainly gives you both higher confidence in any individual measurement and also provides some complementary information where transcripts, for example, may not equate to expression of a particular protein either in the tumor or the microenvironment.
Are there issues with particular samples, for example, the age or an FFPE tissue block?
So there clearly are issues with particular samples. We were fortunate to be able to use this rapid autopsy program for our analysis, and we clearly excluded certain samples that, for example, had severe necrosis within them. But we did show that dating back about 10 years, these samples that were formalin fixed and archived still had very suitable nucleic acid and protein for the DSP-based analysis. And that's something that we wouldn't have been able to evaluate at the, like using a technology such as RNA-seq. We don't know if even older FFPE tissue blocks would be suitable. They very well may be, but we don't know at this point in time. But at least in our case, dating back 8 to 10 years, they were perfectly suitable for DSP. Bone is still a challenge, no doubt. The samples that we looked at overall had lower signal for the transcript profiles, likely due to the processing that needed to be done for decalcification. But there are some methods where you don't need to use as harsh of conditions for decal. And so I would anticipate that those samples would be of better quality even than the ones that we use. So I certainly wouldn't rule out bone as a routine analyte, but they should be considered carefully.
Are there spatial aspects to immune cells in prostate metastasis that may influence tumor behaviors or treatment responses?
Right. So we don't really know at this point whether there is a spatial influence for immune cells in the metastases. That's a very important and very interesting question. I think that has been assessed in other tumor types where you actually do see robust immune responses, for example, to immune checkpoint blockade, for example, in melanoma, bladder cancer, and some lung cancers. The problem is in prostate cancer, we have yet to see consistent, robust responses, with the exceptions of those tumors that have high mutation burdens due to mismatch repair deficiency. So I think once we start to develop cohorts of patients that we do see responses to with ideally a new immune checkpoint blockade strategy or CAR T-cell based methods, that we will be able to assess the role of, or the response of the spatial contribution to treatments. So that's an active area of work. And as I mentioned in my talk, we haven't yet done a rigorous analysis of the peritumoral immune composition, which could be related to prostate cancer phenotype or the subtypes, and potentially to treatment responses. So that's ongoing work that we hope to complete the next 6 to 12 months. So I appreciate your attention. What I tried to demonstrate here and show through the digital spatial profiling is that advanced or metastatic prostate cancer clearly has different phenotypes with spatial influences that may be important in understanding disease progression in response to therapy, and the striking lack of immune cells within these metastases. So this really opens up the opportunity for future work in enhancing immune responses and also taking into account tumor heterogeneity as we consider different combinations of therapies for advanced cancers. So I want to thank all of the my colleagues, the people that actually did the work related to these studies at Fred Hutch, the University of Washington, and NanoString. Again, thanks for your attention.